Hola, hola. Sí, está bien. Ok. Ok, voy a decir 3 cosas en español antes de empezar. Muchas gracias por venir. Como tenemos un invitado internacional, que es la profesora Lappas de la Universidad de Melbourne, todo este día va a ser en inglés, pero la idea es que, como está dentro del contexto de un curso de posgrado, que los estudiantes participen y todo, y que el lenguaje tampoco sea un impedimento. Entonces, si tienen preguntas en español, no se sientan intimidados en preguntar, y entre todos vamos a poder traducir y hacer esto un poco más dinámico. Eso. Okay, so hello everyone. Good morning. Thank you for coming to this symposium. Before to start, I would like to just mention some important things. For example, our safety precautions. The exit. You have one exit here and another exit in your back. So please be aware where are the exits in case that something happens. Hopefully no earthquake, by the way. Just be aware that is the exit, okay? Also, I would like to acknowledge the contribution of the different faculties that allow us to perform this symposium. The Faculty of Pharmacy and also the Faculty of Biological Science here at the University of Concepcion. Also the Fondesit, because thanks to the funding agency, we can also, invite our speakers today as well. Okay, so we're good? Yeah, okay. Acknowledge, exit, all good. So, well, thank you, everyone. As I've mentioned in the flyer, so this symposium is about extracellular vesicle. I lost the pointer already. It's over there. Yeah. Okay, so the symposium is about extracellular vesicle, and the idea is to discuss a couple of points on the extracellular vesicle field. The idea is to discuss, of course, the different type of extracellular vesicle during this day, mainly focusing exosome, and hopefully when you finish the symposium, at least you will know that exists different type of extracellular vesicle, and not all the extracellular vesicle called exosomes. And for that, we have 5 speakers. We have Dr. Marta Lappas from the University of Melbourne. Then we have Dr. Luis Sobreria from the Pontificia Universidad Católica de Chile. And both will talk about their research line mainly in pregnancy, but also some data about exosome on also extracellular vesicle. Then we have, according to our program, we have our coffee break, and then after the coffee break, we have Dr. Andrew Quest from the University of Chile to talk about also extracellular vesicle and cancer. And then we have our lunch break, and afternoon we have 2 more speakers. Manuel Varas from the Universidad de los Andes, who will talk about exosome, extracellular vesicle in the context of ovarian cancer. And finally, Cristian Carvajal from the Universidad Católica de Chile, who will talk about extracellular vesicle in hypertension as well. So you can see it's different topics. We have all a common interest in exosome and extracellular vesicle. So, of course, I would like to just give you a 5-minute introduction about the context of extracellular vesicle and the exosome. And this is a hot topic in the literature. And it's plenty of information about the potential role of this extracellular vesicle, mainly cell-to-cell communication. So I just put this slide to highlight the explosion in terms of the research on extracellular vesicle, okay? But even if we already know that there are different types of extracellular vesicle, we know also that this extracellular vesicle, we have different subpopulation of vesicle and probably different function. It's a lot of gap in terms of the basic knowledge about the biogenesis, the heterogeneity of this extracellular vesicle, the content of this extracellular vesicle. So even if there's a lot of research going on in terms of the extracellular vesicle of exosome, it's a lot of very important basic questions that need to be answered as well. So to just put everything in a context, I have this simple and a little bit fancy video about extracellular vesicle and why this extracellular vesicle is like a hot topic now in the literature. So you can identify extracellular vesicle in general in all the biological fluid and also in plasma, in circulation. And they are a hot topic now because they function as a transporter, like a carrier of information. They are like letters in which you can put information on it, and one cell will release this vesicle, which contains specific information that seems to be a metabolic representation of the originating cells. And then this vesicle will interact with other cells, release the content, and modify the biological function of the target cells. So it's just that is the basic concept about extracellular vesicle. So you have one cell that have the capacity to communicate with other cells, send a specific message inside of this vesicle. This message can be a specific proteins, can be a specific microRNA, because they are enriched in microRNA, and modify the phenotype of target cells. So it's a lot of information, mainly in cancer, about how this exosome can prepare the pre-metastatic niche and prepare the invasion of cancer cells. So in terms of you just do a quick searching in PubMed, you can see this is the number of publications per year, and you can see how is the explosion of this publication in the last 10 years. Okay, so it's a hot topic in the literature. And so what we know in terms of the biogenesis of exosomes. So this is just a cartoon with the cells, and you can see that the exosomes, they are coming from endocytic compartments. And this is the main difference with other type of extracellular vesicle that is coming mainly from the plasma membrane, like microvesicle. So the exosome, they have a different origin. Also, people can separate the vesicle according to the size. There's a lot of information about the size of microvesicle, and they are bigger compared to the exosome. Exosome, the size is more uniform, around 100 nanometers. But I think the size is not that important, and probably the different origin of this vesicle is the main difference between exosome and other type of vesicle. So we know that the exosome can circulate in the cytoplasm as intraluminal vesicle. In some point, this intraluminal vesicle will be incorporated in multivesicular bodies, this structure, and then traffic into the plasma membrane, bind with the plasma membrane, and release this exosome. And we also know it's a lot of information in cancer and some information also in pregnancy, in which seems to be that these cells can sense the microenvironment, for example, and the low oxygen tension, which is a common characteristic of tumor microenvironment, the release of these vesicles are higher, and also the content is different. And also we have some information in diabetes condition in which the exosome, the number of exosomes are higher, and also the content is also different. Then this exosome will be released to the microenvironment and then interact with the target cells. Is different type or different option of interaction, and then release the content and modify the biological function of the target cell. One of the bigger questions in the field is about the targeting also of this vesicle. So this is a cartoon, but in terms of the physiology, when one cell releases exosome, this exosome can interact with many other target cells. So one of the biggest questions in the field is if this vesicle, they already have a predetermined target in which they know what are the organs of tissue that they're going to interact with. And we don't know the answer, but it's a beautiful paper published in one of the big journals in the field, I think it was Nature, in which the author showed that if you change the profile of integrin in the membrane of exosome, this profile of integrin can also regulate the targeting of the exosome. But we don't have much more information about that. If you don't like the cartoon and you want to see a real image, this is a beautiful electron microscopy image about the exosome. You can see the exosome here. This is the plasma membrane. You can see here the multivesicular bodies and the exosome inside, okay? So you can see they are a small vesicle, and then they are released from the plasma membrane. So in terms of the size, you can see that the exosome usually is around 100 nanometer size, and the microvesicle, they are bigger. And it's important also to point out that it's a lot of information, especially 5 or 10 years ago, about the morphology of the extracellular vesicle, and they believe 5-10 years ago they were like a cup-shaped morphology, but we know now that the cup shape is more like an artifact based on the sample preparation for the electron microscopy. So you can do a cryo electron microscopy. You can see they are a circle, the exosome and the other extracellular vesicle as well. So how you isolate the exosome is just a little bit of background. You can isolate exosome from cells that include primary cells or cell lines from the cell conditioned media. You can isolate the exosome, define the content, and also take this exosome, interact with other cells. A couple of big common questions when you isolate exosome from cells is about how many cells you need to get something, enough amount of exosome to perform your experiment. I think that is one of the bigger questions in the field. Of course, the isolation method also is important. Also, you can isolate exosome and other type of vesicle from biofluid, plasma serum. So they are very attractive as biomarker because they are small, so they are very stable in circulation, and they can transport specific information, microRNA and protein. So as the content is a representation of the originating cell, they are very attractive as biomarker discovery. And also you can identify in saliva and other biofluid. Using a similar approach, you can isolate the exosome and then characterize the content and do some bioactivity assays. So what is the key when we work with the extracellular vesicle? Probably is the isolation. Just I would like to point out that the exosome is a specific type of vesicle in which when you are working with the vesicle, also you need to perform at least 3 or 4 different experiments to demonstrate that you are working with the correct number, with the correct type of vesicle. It's just, this is a table, it's published and available online, and you can see the different methods to isolate exosome. And every single method is different, and probably every single method also can isolate different type of vesicle. It's something that you also need to keep in mind. This is a cartoon in which you can see that it's not a perfect method to isolate exosome, and every single method have pros and cons. And probably you can, of course, there are some methods that are very time consuming in which you can have a better purity of the sample, but the yield is lower. So it depends what is your aim. If you're looking for proteomics or profile the content of this exosome, probably the purity of the sample is important. If you're looking for exosome for injecting animals or perform some in vitro experiments, I think it's difficult to get good purity of the sample because probably you're not going to get enough to perform your experiment. So you can see it's a lot of variability in terms of the isolation of the exosome. So the only advice that I have, and hopefully we can discuss this during the day, is whatever method that you're using, trying to be consistent, use the same method all the time. This is just a paper that we published earlier this year about we described 3 different methods to isolate exosome that you can take a look if you are interested. Also, the exosomes are very attractive for diagnostic. So you just have a table in which is available online as well, in which you can see that exosome can be isolated from different biological fluid, and then can be used as biomarker from different pathological conditions as well. Okay. In terms of the use as exosome as therapeutics, biomarker discovery, and also as a therapeutic approach, if you go into the clinical trial website from the NIH, you can see it's around 97 different clinical trials now going on in terms of the exosome. So when people think that it's not much about exosome, well, I think we know enough to perform clinical trials, actually. So it's 97 clinical trials going on in different pathological conditions involve the exosome in the experimental, in the trial design, okay? Also, the exosomes are very attractive for therapy, but of course, you need to check your doses, check what is your model, your animal, also the content to check what is the fed, and the mechanism associated with your fed are important as well. So finally, just keep in mind for the rest of the day that exosomes, they are a specific type of vesicle. They are different compared to the microvesicle. So you can, we have some tools to characterize the exosome and other microvesicle as well to make sure that you're working with the correct type of vesicle. And the exosome, you have the capacity to isolate exosome, for example, coming from tumor cells, you can use as an early detection from different pathological conditions. The cartoon is in the context of ovarian cancer, but also you can use it a drug delivery system in which you can incorporate drugs and other molecules in the exosome and use as potential therapeutics. Also, if you can, as you can get exosome from biological fluid, they are like a liquid biopsy. So you can also track the real-time monitoring system of different pathological conditions using the exosome as well. Okay, so that was a very brief introduction about the exosome, and now I will introduce our first speaker. Okay, first of all, I would like to just thank Marta, who came all the way from Australia, from Melbourne to Concepcion. So thank you for taking the time to be with us. And she has been here for essentially the whole week, so I think it's a great opportunity for a student as well. So Marta Lappas is Associate Professor of the University of Melbourne in Melbourne, Australia. And she's the head of the Obstetrics and Nutrition and Endocrinology group at the Mercy Hospital in Melbourne. She is a worldwide expert in inflammation during pregnancy, mainly in preterm birth and gestational diabetes. She has published already around 180 papers, and mostly in good journals, and obtained funding for more than $4 million in the last 5 years. So thank you, Marta, for being with us, and we're looking forward for your presentation. That's for you. No, you need to just hold this. Hold it? Yeah. And you can put her.
Thank you. Thank you for the invitation to present today. And it's fantastic being in Chile for the last week. I almost feel like I'm a Chilean. So today what I want to do is talk about some of the work we've been doing over the last few years on inflammation in pregnancy, and more recently some of the work I've been doing with Carlos on the potential role of exosomes in pregnancy. But before I do that, what I want to do is introduce the university that I work for. So that's the University of Melbourne. So it's a very big precinct in the city centre. It's the oldest and most prestigious university in Australia. It's got, in addition to the university, it's associated with 8 different hospitals, including cancer hospitals and a few maternity hospitals as well. It's also affiliated with many leading research institutes, including the Peter Doherty Institute, which mainly focuses on immunology. Now, it is, as I said, it's the most prestigious university in Australia, and it's ranked number 32 in the world. So the department that I work for is the Faculty of Medicine, Dentistry and Health Sciences. It's got almost 2,000 staff, and most of these staff are research staff. And it also has lots and lots of students, including greater than 1,300 students who are doing masters or PhDs. There's many different themes associated with our faculty, including ageing, cancer, neuroscience and psychiatry, and the field that I work in, which is women's health. And in terms of women's health, we look at complications of pregnancy, as well as various reproductive health, such as endometriosis and menopause, and it's quite multidisciplinary in scope. There are basic scientists, there are clinicians, and epidemiologists as well. So I work at the Mercy Hospital for Women, which is about 20 kilometres from the main campus. This hospital is quite new, and it's co-located with probably about another 10 different hospitals and research institutes. And this is just our laboratory here. And my group, as Carlos said, is the Obstetrics, Nutrition and Endocrinology group. And I have many, many focuses for my research, particularly preterm birth and gestational diabetes and obesity. And these are just some of the samples that we collect, including placenta, myometrium, decidua, maternal and cord bloods. We also collect adipose tissue and skeletal muscle. We also do some studies in depression in pregnancy, where we're looking at the impact of depression and antidepressants on fetal outcomes, as well as a long-term gestational diabetes follow-up project where we are trying to predict the development of type 2 diabetes in women who've had gestational diabetes. And these are the fantastic midwives that we have who collect all our samples for us. And so our system that we use is it's called REDCap, and when the research midwife consents the patient, she obtains all the clinical information on the mum and the baby in real time, and they are put into the system, so we have all that information immediately. So that's just a bit about our group. And in terms of the talk, my talk, so what I want to do is talk about the pathophysiology of gestational diabetes. I'll then introduce the role of inflammation in gestational diabetes. In particular, I'm focused on the role of adipose tissue in disease pathophysiology. So I'll talk about adipokines and some of the recent work we've been doing on exosomes from adipose tissue, and then how we can prevent gestational diabetes by targeting inflammation. So gestational diabetes is defined as any degree of glucose intolerance with the first onset or first recognition during pregnancy. And what is quite alarming is that GDM is on the rise, and this is in concert with the obesity epidemic. In our hospital, it complicates about 20% of all pregnancies. So it not only impacts the mum, but what is also distressing is that it can have long-term effects for the offspring. So these offspring are usually born bigger, they have increased fat mass and adiposity, and this predisposes them to a number of diseases later in life, including diabetes, cardiovascular disease, and obesity, and also certain cancers. So there's a huge, huge economic cost associated with gestational diabetes. In pregnancy, caring for a woman with gestational diabetes costs about 35% more than it does for other women. Post-pregnancy, 2 to 5 years post-pregnancy, it's also showed an increased costs of about 20%. In addition, there's also the long-term costs of gestational diabetes in terms of both mum and the offspring, and these costs are incalculable. So what is really, really depressing is that the current treatment and preventative strategies for gestational diabetes are really quite unsatisfactory. And insulin, which is commonly prescribed for gestational diabetes, is really fantastic in regulating mum's blood glucose levels. However, it doesn't protect the fetal offspring, adverse effects on the fetal offspring. In addition, there's also some recent evidence to suggest that insulin therapy may also be associated with increased risks of developing type 2 diabetes later in life for the mum. There is certainly some very promising data on diet and exercise in preventing gestational diabetes. However, this is really only effective in high-risk groups. It's also complicated because people have their own certain diet preferences, as well as there's compliance issues. So clearly we need a better therapy to prevent gestational diabetes. However, to do this, we need to better understand the pathophysiology of gestational diabetes. So gestational diabetes is characterised by insulin resistance. Now, insulin resistance is a normal part of pregnancy, and this happens usually in the 3rd trimester, and it's important because what it does, it directs the nutrients away from the mum towards the placenta. The placenta can then transfer these nutrients to the fetus to allow for adequate growth of the fetus. In women with gestational diabetes, however, this insulin resistance is much more severe, which results in increased placental uptake of the nutrients and therefore accelerated fetal growth and development. So our studies over 10 years ago showed that in adipose tissue and skeletal muscle of women with gestational diabetes, the insulin-stimulated glucose transport is suppressed. There's reduced expression of the insulin signaling pathway components, as well as reduced expression of glucose transporters. In concert with maternal peripheral insulin resistance, GDM is also characterised by changes in placental nutrient metabolism. So again, in normal pregnancy, amino acid activity, nutrient transport activity is upregulated, and this again is to allow for optimal fetal growth and development. However, in women with gestational diabetes, this nutrient transport, particularly amino acid system A activity, is increased, leading to higher uptake of nutrients. There's also evidence that there's higher accumulation of fatty lipid droplets in the placenta, and also increased expression of fatty acid transporters, again leading to increased nutrients to the baby. So what we think is that inflammation regulates insulin resistance and and placental nutrients transport, and what I'll do is present some of our evidence. So what we know is that pro-inflammatory cytokines, they increase during gestation. They are predictors of high insulin resistance during pregnancy, particularly adipokines such as TNF-alpha. And they also correlate with fetal adiposity, again, particularly adipokines such as leptin. We know that individuals with gestational diabetes have increased leukocyte counts and also an altered adipokine and inflammatory biomarker profile. Our studies have shown that placenta and adipose tissue and skeletal muscle contribute to this increased inflammatory state by synthesizing and secreting a number of pro-inflammatory mediators, and these are dysregulated in gestational diabetes. So these adipokines, such as interleukin-1 beta, interleukin-6 and TNF-alpha, have been shown to induce maternal and placental inflammation. They can cause maternal skeletal muscle and or adipose tissue insulin resistance, as well as altering placental nutrient uptake, and I'll just show some of that evidence now. So this is some of our data published a few years ago, demonstrating that what we have here is the concentration of the pro-inflammatory cytokine interleukin-1 beta being secreted from adipose tissue. So we have normal glucose tolerant women here, women with gestational diabetes, either diet controlled with insulin, or combined. And as you can see, the secretion of interleukin-1 beta is increased in adipose tissue from women with gestational diabetes. This secretion is dependent on the inflammasome, as we have increased expression of caspase 1 in these same samples, and caspase 1 is involved in the activation of interleukin-1 beta. So then given that interleukin-1 beta 1 was increased in GDM, the next question we asked is, can it cause defects in the insulin signaling pathway in skeletal muscle? So we incubated skeletal muscle with interleukin-1 beta and looked at the expression of insulin signaling pathway components, and found that indeed it could suppress the expression of phosphorylated insulin receptor substrate 1, And this was associated with decreased expression of the glucose transporter, GLUT4, and this resulted in decreased glucose uptake in skeletal muscle. So studies have also shown that cytokines or adipokines can also alter nutrient transport, and these studies are mainly done by Professor Thomas Jansen and his group. And so they've incubated placental cells with, in this case, either TNF-alpha in white or interleukin-6 in black bars, and shown that these cytokines can increase system A amino acid uptake in placental cells. They can also increase fatty acid accumulation in placental cells, and so this is just some data showing that interleukin-6 can increase the expression of fatty acids. So what causes this inflammation? Well, we know obesity is one of the main causes. However, this only accounts for 30% of cases of gestational diabetes. So what are the other causes of the inflammation? Now there's some evidence that infection can be involved in the pathophysiology of gestational diabetes. So bacteria and viruses can activate toll-like receptor signaling to induce insulin resistance and changes in placental nutrient transport. There is some evidence that women with bacteria or virus infection during pregnancy have increased risk of gestational diabetes. In addition, maternal peripheral leukocytes have increased levels of toll-like receptor 4 expression. So the question then we asked is, then can viral products or bacterial products induce inflammation, cause skeletal muscle insulin resistance, and also alter placental nutrient transport? So what we did here was to look at the effect of a viral product, poly-IC, and that activates TLR3 signaling pathway, and found that incubation of adipose tissue and placenta with poly-IC could increase the expression and secretion of pro-inflammatory cytokines from these tissues, suggesting increased inflammation. We then looked at the effect of poly-IC on the skeletal muscle insulin signaling pathway, and again found that incubation with this TLR3 ligand could suppress the insulin signaling pathway components, GLUT4 expression, and then glucose uptake. We then looked at the effect of viral and bacterial products on amino acid transport in trophoblast cells, and again found that these products could, so we've got here TLR3 signaling, and this is LPS, which is TLR4 signaling, found that incubation of our placental cells with viral or bacterial products could increase the expression of amino acid transporters and amino acid uptake. So that's our work on inflammation. But in addition to secreting adipokines, adipose tissue also secretes exosomes. Now, Carlos has spoken quite nicely about exosomes, but what I want to stress is that exosomes are really quite exciting because they are representative of the cell that they came from, and then those exosomes can then travel elsewhere, release their cargo, and cause defects in the other tissues. With respect to exosomes and gestational diabetes, Carlos and his team have done the majority of the work in this. And what they found is that placental exosomes under hypoglycemia or gestational diabetes, the release is increased. There's also increased levels of exosomes in maternal plasma of women with gestational diabetes. And interestingly, the pathways that they target are energy production, inflammation, and metabolism. However, what was quite interesting was they were not attributed solely to the placenta, which is thought to be the main source of circulating inflammation. So they weren't attributed to the placenta because the ratio of placental exosomes in GDM was actually lower. This suggests that there are other tissues that may be contributing to these increased exosome content in women with gestational diabetes. So it's possible that adipose tissue derived exosomes may also be contributing to these exosomes. So therefore, our overriding hypothesis is that adipose tissue exosomes from women with gestational diabetes carry a specific set of proteins, and these may influence placental function. So to test this hypothesis, what we did, we took adipose tissue from 140 pregnant women at the time of term cesarean section. We had 70 with gestational diabetes and 70 normal glucose tolerant women. We did adipose tissue explants, we collected the media, and then exosomes were isolated according to methods optimised in Carlos's lab. So firstly, what we did was to characterise the exosomes, and as you can see, the majority of the exosomes are in the 100 nanomolar range. Exosomes were also characterised according to the presence and absence of exosome-specific markers. So what we have here is 3 specific markers of exosomes, CD63, CD9, TSG101, and they were present in the exosomes. And here we have GRP94, which is an endoplasmic reticulum protein, and this was absent in the exosomes, showing the purity of our extraction. And again, we have the shape of the exosomes. So what was quite interesting is when we compared the profile of normal glucose tolerant women compared to women with GDM, we found an increased content of exosomes in adipose tissue, in exosomes derived from adipose tissue. Now this is quite interesting actually because, as I said before, women with gestational diabetes have increased circulating levels of exosomes which are not due to the placenta. So this data suggests that the increased content of exosomes in GDM women may be coming from adipose tissue. We then looked at the relationship between adipose tissue-derived exosomes and clinical markers of or clinical characteristics of the patients. And what we found, which was very, very exciting, was a significant and positive relationship with exosome content and fetal birth weight. Now, this is really exciting because, as I said, fetal gestational diabetes is associated with offspring with increased fetal birth weight. So this suggests that exosomes from adipose tissue may be a link between placenta and nutrient growth. We then looked at the protein profile of the exosomes from adipose tissue, and here's a volcano plot, and of the there was 127 proteins that were significantly different between the two groups. There were 110 that were upregulated in the GDM exosomes and 17 that were downregulated. And what was quite interesting is that the pathways that were associated with these were involved in cell and nutrient metabolism, and they included those involved in mitochondrial dysfunction, mTOR signaling and sirtuin signaling pathways, and we know that these pathways are involved in not only inflammation but also in nutrient metabolism. So then the next question we asked is, do adipose tissue-derived exosomes also regulate genes associated with glucose metabolism in placental cells? So to address this, we took adipose tissue-derived exosomes from normal glucose-tolerant women and women with gestational diabetes, and we added these to placental cells. We did a glucose metabolism PCR array and found that there was probably about 45 genes that were differentially regulated in the two groups. And quite interestingly, the pathways that were the most significantly regulated were those involved in glycolysis and gluconeogenesis, again demonstrating the link between adipose tissue-derived exosomes and fetal growth. So this is our working model. In gestational diabetes, you have sterile inflammation, bacterial and viral infection. This activates the adipose tissue to secrete adipokines and exosomes. They can cause maternal peripheral insulin resistance, leading to increased levels of nutrients in maternal blood. They can also increase the expression of nutrient transporters in the placenta. The placenta can then take up these nutrients, leading to increased babies with increased fetal weight, and then leading to the long-term effects of gestational diabetes. So we think that inflammation, particularly from adipose tissue, is quite central to this process. So the next question we asked is, can we target inflammation to prevent gestational diabetes? So I just want to briefly talk about some of the work that we've been doing on polyphenols. So polyphenols are bioactive components found in fruits and vegetables and nuts and seeds. And we've known for a long time that a diet rich in these foods is very protective. There's also some evidence to suggest that a high intake of the plant diets in pregnancy is also associated with improved pregnancy outcomes, including gestational diabetes, and there's some evidence using Mediterranean diet and the DASH diets, and these are diets high in these fruits and vegetables. Some small trials in gestational diabetes have shown that they can improve levels of insulin, inflammation and oxidative stress. However, we don't know what contribution polyphenols have to these positive effects. So that was an aim of our project. So we are doing, there's two ways we are assessing this, and that's using in vitro and in vivo model of GDM. So in vitro, we take placenta, adipose tissue and skeletal muscle from pregnant women. We use TNF-alpha to recapitulate inflammation and the defects in the insulin signaling pathway associated with GDM. And in our in vivo mouse models, we use two mouse models of gestational diabetes. One is the heterogeneous leptin receptor gene knockout model, and that's a model of insulin resistance. And we use an adiponectin gene knockout mouse model, and that's a model of beta cell dysfunction. Now, these models are quite neat because when these mice become pregnant, they spontaneously develop gestational diabetes. In addition, the offspring have also got increased weight, which are the maternal and fetal characteristics of gestational diabetes. So then what we've done is to use various polyphenols to see their effects on the pathways involved in gestational diabetes. So here we've got some data in skeletal muscle incubated with two different polyphenols, nobiletin and naringenin, and they are primarily found in citrus fruits. And so what we can see here is we've got a Western blot showing that TNF-alpha, as expected, decreases the expression of the GLUT4 in skeletal muscle, and this effect can be alleviated by pre-incubation with polyphenol nobiletin. Here we've got glucose uptake in our skeletal muscle, again demonstrating that pre-treatment with our polyphenols can alleviate the decrease in glucose uptake mediated by TNF-alpha. In vivo, so these are in our mouse model, this is the leptin receptor deficient model. We have from gestational day 10 to gestational day 18, the mice receive oral gavage of our polyphenols, and then we do on gestational day 17, we do an oral glucose challenge test to look at glucose levels. And what we have here is fasting glucose, and this is the area under the curve after the glucose challenge test. And as you can see, treatment with our polyphenols significantly decreased fasting glucose levels as well as the area under the curve. We then also looked at inflammation. So this is some of our in vitro studies in placenta and adipose tissue. So as expected, TNF-alpha increases expression and secretion of pro-inflammatory cytokines, and pre-treatment with nobiletin or naringenin can significantly suppress this effect. Likewise, In our mouse model, we have placenta, visceral fat, and subcutaneous fat here. And again, you can see this is, I think this data is representative of a couple of cytokines and chemokines, such as interleukin-1 alpha, MCP, showing that pretreatment with our polyphenols can suppress the inflammatory profile in the mice as well. So, in conclusion, we know that there's increased release of adipokines and adipose tissue-derived exosomes from women with gestational diabetes compared to normal glucose-tolerant women, and these can positively correlate with fetal birth weight. Adipokines and adipose tissue-derived exosomes may mediate the metabolic communication between maternal adipose tissue and the placenta. I've shown evidence that they can induce maternal and placental inflammation. They can cause skeletal muscle and adipose tissue insulin resistance, and they can also alter nutrient uptake. I've also shown some evidence that anti-inflammatory agents such as polyphenols may be promising and novel therapeutic for preventing gestational diabetes. I would just like to acknowledge some of the researchers, particularly the clinical research midwives who collect the samples for us, and the patients who donate all their samples, and all the various funding bodies. Thank you.
Thank you, Marta. I think it's quite a fascinating story, especially the exosome part. So we have time for questions. Probably can start with probably the easy question now, your story about polyphenols. So the amount of polyphenol that you add into your in vitro and your in vivo system, they are similar to, for example, that the concentration that we can find in food, for example. Meaning you can eat whatever, a couple of vegetables or something like that. Yeah.
So it's quite hard to quantify the level of polyphenols because each apple is different in terms of the polyphenols that they'll have. So there's certainly some evidence to suggest it's about, I think, 500 milligrams is about the average that you might get in a diet. And when we compare the doses that we're using in mice, which is 50 mg per kilo, we're probably using a little bit. We're probably using 300 to 500 mg. So it's quite comparable in terms of the concentrations. However, it's hard to quantify really what a person eats.
According to the same question, what kind of polyphenols are you using, or where's the origin of the polyphenols that you're adding to?
So these are just chemical compounds that we buy, like from Sigma.
So you don't?
We don't extract
you don't
them.
insulate?
No. I don't extract the polyphenols.
Okay.
So the whole aim is to be able to, in terms of a therapeutic, you want something that's very cheap, that's easily available, that you can just give in a pill form. That would be the hope. So we wouldn't be extracting it from an orange. You would need a lot of oranges. So
as vitamin C? No. It's another question. You don't have proof any property of different fruits or vegetables that have different concentration of polyphenols and different kind of polyphenols in your model.
So we've looked at 2 from citrus fruits here, but we're looking at various polyphenols. So they're just 2. So we've got another 7 or 8 that we're looking at different types from different foods.
Yeah, nice talk. So I was just wondering, so you basically showed that bacteria, probably the LPS somehow, affects this system. In China, there's a very high prevalence of Helicobacter pylori. So I was wondering whether, do you know of any kind of connection there between gestational diabetes and maybe Helicobacter pylori?
No, I don't know if there's any relationship. That's quite interesting. I don't know if there's any relationship, but it certainly fits in with.
Right. Exactly. And the other question I had, I don't know whether you thought about this, but bacteria also produce outer membrane vesicles. So I'm just wondering whether it's actually the LPS or the outer membrane vesicles that might be.
Good point. I don't know. That's a good point. Yeah.
Might be worth looking
Yeah,
into.
maybe worth looking at. Yeah, absolutely.
Thank you, Marta. Just a quick and very simple question, I think. Your exosomes from the adipose tissue were from the obese woman or overweight woman,
So
or a
we
mix?
had a combination. So we had lean, overweight and obese, but they were all matched for BMI. So our two groups didn't differ in terms of their BMI.
Right,
So we had a mix because there were 70 patients in each group, so we had a mix.
and do you think it will be a difference between overweight and obese
We found no
release
relationship.
exosomes?
Yeah, when we looked at the relationship, there was no effect of BMI on content of adipose tissue, of exosomes, sorry.
You mean the content, but you mean the cargo of the exosomes, the number?
Numbers,
Ah, right.
yeah.
But then
Yeah.
the cargo may be different, not just the number, I think.
Did we look at the car?
I don't remember, but I think that's a good point actually
Yeah.
in terms of the VMI. And the issue that we didn't find any correlation between the number of exosome and maternal VMI
Yeah.
was quite a surprise actually. So, and the number we didn't find difference. And for the proteomic analysis,
We didn't separate according to
we didn't
BMI.
because we didn't find any
Yeah.
difference in the concentration. Yeah, but it was very, very interesting actually because previously we already published a paper in which we identify difference in the maternal BMI and the number of circulating exosomes in plasma. So when we performed the experiment, we hoping to get a similar difference, but we didn't find
Yeah,
it.
and we had enough numbers in each group,
Yeah.
so it wasn't a lack of numbers.
And the other important issue when we work with patients, because this is biological sample coming from independent patients, we have a lot of variability also in the groups. So it's something that we need to keep in mind when we perform the analysis.
A very nice talk. Thank you very much. So I have like a question related with the immune system. So during pregnancy, you know, it's a very anti-inflammatory environment for the immune system. So we're supporting like an anti-inflammatory environment. But then you show that in this case, you have pro-inflammatory cytokines, probably from tissue as well. So I was wondering if you think that the immune system is like sensing these cytokines and said, oh, it's a lot of inflammation, I need to like downregulate this, or the immune system is like playing like a bad role and then said, oh, we have interleukin-1, we're going to differentiate to a pro-inflammatory Th17, for example. Do you have any idea about that?
Well, I think as pregnancy progresses, inflammation increases. So that's been, like in terms of looking at inflammation, it does increase as gestation increases, and that's obviously to allow, cytokines can be good and bad, but I think in that situation it's obviously a good thing, and we think it does that because it directs nutrients away from mum towards the placenta in terms of the studies that we're doing. So I think that what you're probably talking about is early in pregnancy where it's quite an anti-inflammatory state. Is different to as pregnancy progresses, it becomes pro-inflammatory. And it becomes pro-inflammatory not only for that, but it's an inflammatory state. In order to prepare for labour and delivery, you've got to have inflammation. Inflammation seems to drive labour and delivery, so I think as it progresses, it definitely becomes pro-inflammatory.
Okay, any other question?
Very nice talk. I was wondering about the contents of the exosomes when you did the proteomic. This is from the total exosomes or is a subpopulation?
It's from the total exosomes.
Total exosomes.
Yes.
Okay, probably there are more than one kind or type of exosomes.
Carlos is the expert. As far as I know, there's
sorry.
only one type of exosome.
Yeah.
So this particular exosome coming from adipose tissue explant. So essentially, probably in the explant you have a mix. Probably you have macrophages, you have adipocytes, and you have different types of cells in the explant. So when we get the exosome from the cell condition medium for this plant, we just call total exosome from this plant. But you're totally right. That can be, we don't know if it's coming from the macrophages or other types of cells.
Is that what you mean? Different,
Yeah.
different
I guess, yeah.
cells in the tissue contributing to
those
the different-
exosomes.
Yes,
Okay.
That's
you
a
would
good
have
question.
to obviously separate
Yeah.
the different compartments of the cell, of the tissue. But the issue with that is we just don't get enough tissue to be able
Yeah.
to separate
Yeah, I
the
know.
different populations.
Yeah, I know it's an issue.
Yeah.
That's the reason I asked.
Yeah.
If you have any way to separate them, I don't know, cell sorting or beads, magnetic bead with a specific marker for subpopulation of exosomes.
Yeah.
I don't
So,
know.
yeah,
Yeah, that's
look,
possible.
yeah, I think that is a very good comment. But
Yeah.
also, at least for the aim of the research, we already identify an effect of exosome coming from the adipose tissue, and then we provide the content.
Mhm.
So now we need to identify what are the mechanisms associated with the effect, and then probably focus in the microRNA or the protein that are in the exosome.
Yeah.
And that is very interesting approach because everyone working in extracellular vesicles seems to, they identify effect first, and then they identify what in the exosome drive the effect on the target cell, and then they focus in that molecule, and the exosome of the vesicle become a little bit irrelevant. And probably that is the best strategy to continue further with the research. Otherwise, you will have the problem about if you have the same content in every single exosome isolation.
Thank you.
Okay. Yeah. Thank you, Marta. Yeah. Okay, fantastic. So we just move forward to our next speakers. So our next speaker, he doesn't need much introduction for us because he was an academic here in our institution before.